Hello students, today we are going to start with the reproductive physiology. Hope you are all keeping well and safe at your places. This is the only means of communication by which we can take your classes at this point of time. So I'll be uploading some videos. Look it up and consult your textbooks, and then we'll have some interactive classes where we can clear your doubts. So let me start with the first topic of reproductive physiology, that is the sex determination and sex differentiation. Let us share the screen. So as we go through the chapter, you'll be able to explain how bipotential tissues are directed to differentiate into male or female gonads, and how the embryonic ductal system, that is the Mullerian or the Wolffian system, develops into the female or the male internal sex organs, and how development of external genitalia, which is a hormonal regulation, can determine the phenotypic sex, and we'll discuss certain abnormalities of sex differentiation. Now, sex determination in a fetus is determined by the genetic makeup of the individual, that is, its chromosomal makeup. If it is 46XX, then it's a female individual, and if it is 46XY, then it is a male individual. And the sex chromosome plays a very decisive role in this sex determination. So this is the Y chromosome present in male individual, the XY individual, and this is the short arm of the Y chromosome, where in 11th position we see a gene is located which is known as SRY gene, or the sex determining region of Y. And this gene actually determines the development of testis in male individual. So if this is present, then an individual develops into male, otherwise the individual will going to develop into female by development of ovary. Now, once the sex is determined as early as 6th to 7th week of intrauterine life, or rather we can say 7th to 8th week of intrauterine life, then comes the question of sex differentiation. That is, there occurs a multiple process which ultimately differentiates from a zygote into the distinguishable male or female characteristics. It is definitely the chromosomal sex which guides it, which develops into gonads, and the hormonal secretion from gonads ultimately differentiate into internal and external genitalia. So the sex differentiation is a complicated multiple processes involved in development of distinguishable male or female characteristics. And this sex differentiation occurs at 3 levels: the chromosomal sex, the gonadal sex, and the phenotypic sex. Now, once chromosomal sex is determined from the parent, either the XX, that is female, or XY, which is male, and here we know that father is the decisive figure who will contribute Y chromosome to give birth to a male individual, or X chromosome to give birth to the female individual, because mother will be contributing X chromosome to both of them. Now, those who have the Y chromosome or the SRY region, they will secrete a cascade of factors, of which one is very important, that is the testis determining factor, and this will help in differentiation of the bipotential gonad, the medulla of the bipotential gonad, into testis. And this is actually the gonadal sex. If SRY is absent, then the bipotential gonadal cortex will develop into ovary, and so the bigonad, it is the female which is going to develop in utero. Now, once testis is developed, we see that subsequently in early intrauterine life, there is secretion of the hormone testosterone from the Leydig cells. And this leads to the development of male genitalia from the immature ones. And testosterone, by presence of 5-alpha reductase, forms 5-alpha dihydrotestosterone or DHT, which actually helps in development of the external sex characteristics. And this entire hormonal regulation occurs between 8 to 13 weeks of intrauterine life, and by 16th week of intrauterine life, the phenotype is determined. And it is due to the presence of the Mullerian regression factor or the Mullerian inhibition factor secreted from the Sertoli cells in intrauterine life that the Mullerian system destined to develop the female genitalia degenerates. So, in case of male, the phenotypic sex is developed under the influence of the hormone testosterone, dihydrotestosterone, and the Mullerian inhibiting factors. In case of female, on the other hand, it is the default setting that if these hormones are absent, then the female genitalia, both internal and external, will develop and the phenotypic sex will develop into a female. And finally comes the metabolic and the behavioral sex. This completely develops at puberty when there is enough of the estrogen and progesterone in female, and male have enough of testosterone and dihydrotestosterone, and finally the adult phenotype is developed. So right from the intrauterine life till puberty, there is the complete sexual differentiation to develop into an adult male or female. Thus, the sex development, as we have already discussed, has 3 components: the chromosomal components depending on the karyotype, and there may be several variations. For instead of 46XX, we may have 45X0. We may have 47XXX, triple X, or XYY. So there are several chromosomal abnormalities which we can come across in the sexual development. Then the gonadal sex, the development of ovary or testis. But there are many individuals who have ovotestis, both of them present at the same point of time. These are called the true hermaphrodites. And then the phenotypic or the anatomic sex, depending on the hormonal regulation, which gives rise to the development of the internal and the external genitalia. And here also there may be several abnormalities giving rise to ambiguous genitalia, depending on the hormonal exposure of the individual. And we find that there may be pseudo hermaphrodite, both male and female, depending on this. Last but not the least is the psychosexual development or the brain sex development, which occurs right from the intrauterine life due to the exposure of the male individual to the brain, to the testosterone, and subsequently during puberty when the male and female hormones are present. And not only that, the several biologic factors, the environmental factors, and the social influences develops the gender identity. And often there is a discordance between the organic sex and the psychological sex. And these are usually the transgender. So gender identity is also a very important phenomena in the individual life. So we see that the genetic sex is determined from the parents, and then under the influence of Y chromosome, the gonadal sex is developed, that is testis in case of male, and in the absence of SRY, the development of ovaries. Now, once the testis is developed in intrauterine life, we see that the hormonal production, and that influences the brain in the intrauterine life itself. And then definitely under the hormonal influence, there is development of internal and external genitalia. And so a child is assigned or born with a specific sex, male or a female, and subsequent rearing occurs like that. They have the male or female outfits given. They are given the toys this time for the male or female. There is differentiation, and so there is similar rearing according to the gender assigned. Subsequently, puberty comes, so the hormone production at puberty, because female hormone mainly produced during puberty, they also helps in the differentiation of brain and definitely development of the secondary sexual characters, and this finally assigns a one with a complete gender identity. And here, if any discordance occurs between the organic sex and the psychological sex, then the gender identity becomes abnormal. Now, so we are coming back to the question of who determines the sex. Sex is, as we have already told, determined genetically, and it is the Y chromosome, the 11th position, where SRY gene is present, and this encodes several transcription factors, like one we have already mentioned is the testis determining factor. This TDF initiates a cascade of transcription factors to develop, like SOX9 protein, then steroidal factor 1, then definitely from Sertoli cells, the anti-Mullerian factors, then MAP4K1. These all helps in the male differentiation of sex. The ovarian development, as we have said, is a default setting, but here also certain transcription factors like WT4, then FOXL2, BMP15, etc. are being implicated. So let us come to see how the regulation of male development occurs by the SRY gene. So in male embryo, in the 11th position of the Y chromosome, there is SRY gene, which initiates formation of several transcription factors like TDF, then SRY-POX, then SF1, so on and so forth. So around 6 to 7 weeks of intrauterine life, in the presence of testis determining factor, the testis develops. That is the medulla of the bipotential gonad develops into testis. And once this testis develops, then by 8th to 9th week, we see that the Sertoli and the Leydig cells starts secreting the testosterone and the MIF. Now, this Mullerian inhibiting factor or Mullerian regression factor, this we know that it inhibits the Mullerian system, which is destined to form the female internal genital organs. Whereas the testosterone helps in differentiation of the Wolffian duct to the male accessory sex organs like epididymis, vas deferens, seminal vesicle, etc. So at birth, we find that there is differentiated Wolffian duct into the male accessory sex organs. Similarly, this testosterone in the presence of 5-alpha reductase forms the dihydrotestosterone, and that develops the genital folds and genital tubercle into male external genitalia, that is the penis and the scrotum. And under the influence of testosterone and by the aromatase, the estradiol, which is present in male individual, and definitely at puberty, the pubertal hormone, they subsequently develops into, helps in brain development, and so the male gender identity, male behavioral pattern, and the tonic GnRH secretion, which differ in male and female, occurs at this point of time. So this is how the male development is being regulated by the SRY gene. So let's come to the development of the internal reproductive organ. So up to 6 weeks, the fetus has the bipotential gonads. So these are the bipotential gonads, and surrounding it, this is the mesonephric duct, or also called as the Wolffian duct. This is the mesonephric duct or the Wolffian duct, which is destined to develop male internal genital organs, and just lateral to the mesonephric, there is the paramesonephric duct, which is also known as Mullerian duct, destined to form the female internal genitalia. And this is the metanephros with the urethra, which is going to develop into the transient kidney. And this is the cloacal opening, which is the common opening for the urethra for the reproductive system as well as the anus. Now, after 6th week of intrauterine life, we see that the SRY gene starts producing the several transcription factors. And as we have known that by under the influence of TDF, that this testis, the medulla, which develops into the testis, and this starts between the 7th to 8th week of intrauterine life, that the male testis develops. And then we find that in absence of SRY gene during the 7th to 8th week of intrauterine life, the cortex, the gonadal cortex, the bipotential gonadal cortex, it develops into the female ovary. And subsequently, we see that once the testis is developed, the Sertoli cells secrete the anti-Mullerian factor, which helps in degeneration of the Mullerian or the paramesonephric duct. And so now the Wolffian duct only survives. And this, under the influence of the testosterone and subsequently dihydrotestosterone, develops into the epididymis, into the vas deferens, and into the seminal vesicle. And it is the prostate which develops from the urogenital sinus. Not only this, but by 13th to 16th week of intrauterine life, we see there is descent of testis into the scrotum. So everything occurs in the intrauterine life under the influence of the hormone testosterone and dihydrotestosterone. Now by 10 weeks, if there is no SRY gene and no testosterone, then the Wolffian ductal system gradually degenerates and the Mullerian system takes the upper hand. And it is the default setting that without the SRY gene, without the testosterone hormone, and definitely without this Mullerian inhibiting or anti-Mullerian hormone, we find that the paramesonephric or the Mullerian duct develops into the fallopian tube, the uterus, and the upper third of vagina. So female, we find, is an default setting which develops into ovary, the bipotential gonad, and the Mullerian duct into the internal genitalia between 10th to 16th week of intrauterine life, if it is not intervened by the SRY gene secreting several transcription factors, including TDF and also the anti-Mullerian factor, which is inhibiting the Mullerian system. And then it is under the influence of the hormone, the testosterone and the dihydrotestosterone, the internal genitalia, they develop. So what are the homologous organs which develop from the Wolffian duct, from the urogenital sinus and the Mullerian duct? In case of male, it is the Wolffian or the mesonephric duct, which actually gives rise to the internal sex organs, the epididymis, vas deferens, and the seminal vesicle. Whereas in female, it is the degenerated epiphuron and the Gartner's duct. In case of female, on the other hand, Mullerian duct takes the upper hand, developing into fallopian tube, uterus, and vagina. And in male, they are the appendigeal organs like appendix of testis and the prostatic utricle. Urogenital sinus helps in development of bladder in both sex, and prostate in male, and the distal vagina, parts of distal vagina in case of females. Now comes the development of the external genitalia. This starts developing after 10th week, but by 16th week, the external genitalia also gets fully developed. Now, in the indifferentiated state, we find there is a genital tubercle with the urethral folds and the urethral groove, and this is the labioscrotal swelling, and just behind is the anal opening. Now, in case of male, we find that the genital tubercle, under the influence of the hormone dihydrotestosterone, develops into glans penis, and it is the extension of the genital tubercle which gets folded with a just a remnant of the urethral fold, and this is called the shaft of the penis. And there is the fusion of the labioscrotal swelling also to form the scrotum, where by 16 to 23 weeks there is the descended testis. So there develops the glans penis, the shaft of the testis and the scrotum. In female, on the other hand, genital tubercle remains rudimentary as glans clitoris, and it is the urethral fold which develops into labia minora, having the vaginal opening in the urethral groove, and the labioscrotal swelling, it develops into labia majora, and there is no fusion at all. And subsequently, we find that the anus shifts backward, and there is the complete development of the external genitalia. So if we summarize as this point of time, then we find it is from the parents that the chromosomal sex or the genital sex is being determined, either 46XY or 46XX. Then it is under the influence of the SRY gene that the male testis starts developing. But before that, it is under the steroidal factor 1 and WT1 that the coelomic epithelium and the mesoderm develops into the gonadal ridge. And from here, it is the SRY produced transcription factors like SOX9, MAP4K1 or TDF that the testis develops in male individual. This develops into the Sertoli cells secreting anti-Mullerian factor, which inhibits the Mullerian system, which regresses, and it is the Leydig cells which secrete the androgen, that is the testosterone and subsequently dihydrotestosterone, which helps in the proliferation of Wolffian duct into epididymis vas and seminal vesicle, and the urogenital sinus, the genital tubercle, and the folds into the male external genitalia. Female, on the other hand, in absence of SRI gene and in presence of certain transcription factors like WNT4, FOXL2, and BMP15 and several others, develop into the cortical portion develops into ovary, and the hormone is not secreted in intrauterine life. It is in absence of the anti-Mullerian factor that the Mullerian ducts, they develop into fallopian system, uterus and the upper vagina, whereas Wolffian duct degenerate because of the absence of testosterone. And subsequently, there is also development of the external genitalia, because here no fusion is required. Almost the entire genital tubercle and the urethral folds remain, only the genital tubercle is rudimentary. So this is how there is the sexual determination and differentiation. Now we'll come to certain abnormalities like sex chromosome disorder. Now, we know that in the sex chromosomal disorder, we have one very common syndrome, which is called the Klinefelter syndrome, where the chromosome is XY was there, but another X was added to it. So these individuals, we have male genitalia, but they are somehow not reproductive, reproducible. Okay. Now then comes the Turner syndrome, where the chromosomal abnormality is X0. So only one sex chromosome is present, and that is 45 total chromosomal pairs are present, and this is called the Turner syndrome. Now there are other dysgenesis like X0XY together, non-disjunctions, mixed gonadal dysgenesis. These are having both the potentials, but they have usually do not properly develop either into male or female genitalia. Then there may be XYY. Here, because of the more Y chromosome, these male are tall in stature, normal male, but they have impulsive behaviors, or often the criminal behaviors are common. Triple X, in female, it is usually not decisible, but at puberty we see sometimes they become infertile. And in XXXY, if both are present due to non-disjunction, then these will give rise to the true hermaphrodite. true hermaphrodite having the ambiguous genitalia at the puberty, and they have both ovotestis. And it is the Cheryl Chase, who was the founder member of the INTERSEX society in North America. She is an individual with ovotestis. There are several individuals. These are the intersex phenomena. We'll discuss it. Now coming to the KLINEFELTER's syndrome, 47XXY. It is one of the most common chromosomal abnormalities, 1 per 500 male birth. And because of the presence of Y chromosome, they develop both the internal and external organs that of male. But due to presence of another X chromosome, there is usually dysgenesis of testis, and there is inadequate testosterone hormones. So at puberty, there is feminization of the external genitalia, and most of them are sterile. So they are not reproductively active, and they mostly have some language or learning problems, and testosterone therapy may improve them, having many physical signs. In Turners, we see that the sex chromosome is not paired, only one X chromosome is there, and this is not very common, 1 in 2000 female birth. They have typically short statured, low set ears, and webbed neck. And girls experience the gonadal dysfunction, amenorrhea and sterility, and the coronary heart disease, diabetes, auditory visual problems are common in them. Now, this is the chromosomal abnormalities we are discussing. That is due to the presence or absence of extra or absence of any sex chromosome. Besides that, there may be certain hormonal abnormalities, as we know that in male fetus, mainly under the influence of the testosterone or dihydrotestosterone, and the male internal and external genitalia develops, and it is the Mullerian inhibiting factor which inhibits the Mullerian system. So if there is defective testicular development, so that the testosterone and DHT is not enough, or if there is androgen resistance, then this organic males, that is genetic and gonadal males, may have female internal or external genitalia because there is not enough of testosterone or dihydrotestosterone to develop into male external genitalia. So by default, they will develop the female external genitalia, and these are called the male pseudo hermaphrodites, or this phenomena is called male pseudo hermaphroditism. In case of females or genetic females, organic females, we see if they are exposed to androgen from any source between 8 to 13 weeks of gestation, then there may be development of the male external genitalia and male accessory organs. So they will give rise to the phenomena of female pseudo hermaphroditism. So this is the phenomena of intersex. That is an individual in whom there is discordance between the chromosomal, gonadal, and phenotypic, or the gender identity can be identified as intersex. So what is intersexuality? Discordance between any two of the organic sex, either chromosomal and phenotypic or gonadal and phenotypic, then they are intersexual. But what is transsexuality? Discordance between the organic sex, any one of the factors, along with a psychological sex. That is, they identify themselves psychologically as belonging to opposite sex to that which they belong as the organic sex. So these are termed as transsexuality. So abnormal sexual differentiation due to the hormonal abnormality is true hermaphrodite is definitely not an hormonal abnormality. It is the chromosomal abnormality we have said. The ovarian and testicular tissue both are present in them. But the female pseudo hermaphrodite, where their organic sex is and the chromosomal sex is female, that is, they are having the ovaries, but they have masculine external genitalia, they are the female pseudo hermaphrodites. Whereas male pseudo hermaphrodites are those who have the chromosomal 46XY, they have testis as their gonad, but they have their genitalia developed as that of female. So these are known as male pseudo-hermaphrodite. So true hermaphrodite, we have already discussed. They have both ovarian and testicular tissues. They have ambiguous genitalia because depending on the amount of testosterone which they are exposed between 8 to 16 weeks. And we have said that already there are intersex society in the North America which cater with these intersex people. But the hormonal abnormalities may lead to female pseudohermaphroditism and male pseudohermaphroditism. So here we find that there is certain causes, some causes of this pseudohermaphroditism. In female, we have the congenital virilizing adrenal hyperplasia. Now, congenital adrenal hyperplasia is certain enzymatic defect in the adrenal gland. Now, if we see this steroidogenesis in adrenal cortex, then we find that there are several hormones, and this is the line which develops into corticosterone, then cortisol, and this is the final formation of the androgens and the testosterone. So, if there is deficiency of either cholesterol dismolase, very rare to survive, then 3-beta hydroxysteroid dehydrogenase, 3-beta HSD, or 21-beta hydroxylase or 11-beta hydroxylase, then these will subsequently not allow the vertical steroidogenesis. That is, if 3-beta HSD is absent, then there will only the pregnenolone and 17-hydroxypregnenolone will be produced and not the subsequent congeners. So here they will all go to produce more of DHEA. If there is 21-beta hydroxylase deficiency, there is more production of androstenedione, and from there testosterone and estradiol. Same with the 11-beta hydroxylase. So these hormones, enzymes, if they are deficient, then they will lead to virilizing adrenal hyperplasia. Hyperplasia because if cortisol and corticosterone is not produced enough, then ACTH will be in excess. And if there is deficiency of hormones like 17-alpha hydroxylase or 17-20 lyase, then we find that the vertical lines will be completed, that is corticosterone or cortisol will be produced, but there is no production of the androgens. So in this case, these are the feminizing tumors. So we see that 17-alpha hydroxylase deficiency will cause the feminizing tumor, whereas congenital virilizing hyperplasia will lead to female pseudohyperphroditism. Other cause of female pseudohyperphroditism is male androgen excess, virilizing ovarian tumor like the fibrothecoma or granulosa tumor, and iatrogenic treatment with androgens. And in male pseudohermaphroditism, besides this 17-alpha hydroxylase deficiency, there are two important causes. One is androgen resistance. Even if the receptors are present, there is androgen resistance. And one is defective testicular development from the intrauterine life, so that the testosterone and dihydrotestosterone is not produced enough to produce the male internal and external genitalia. So they have the female external genitalia, although chromosomally and gonadally they are male individuals. So this is the sexual aberrations which occurs in these individuals. So by this I end my class today, and next we will take the class on puberty. Thank you everybody.